Welcome to session 16 of biotechnology LMS paper 2 biochemistry. Today in our session we'll be discussing about the properties and classification of proteins. So the properties of proteins are uh, solubility forms colloidal solution instead of a true solution in water because proteins are large size, because it is due to the large size of the protein molecules. Molecular weight depends on the number of the amino acids. Shape, they have wide variety of shapes. They can be globular, they can be oval, they can be fibrous. Globular example is insulin, oval albumin, fibrous it can be fibrinogen or keratin or something like that. Acidic and basic depends on the ratio of depends on the acidic and basic depends on the ratio of lysine plus arginine is to glutamic acid plus aspartic acid. Uh, Ratios greater than 1 is basic and vice versa. Less than 1 is an acidic. Charge proteins are isoelectric in nature. Proteins are isoelectric. Nature of amino acid determines the pH of the protein. Acidic amino acid like aspartic acid, glutamic acid, and basic amino acid like histidine, lysine, arginine determines the charge on the protein. At isoelectric pH, the protein exists as a dual charged or dipolar ion, which is called as Zwitter ion, and it is electrically neutral, and it is minimum soluble and maximum precipitatable. Least buffering capacity. So these are some of the important properties of proteins. Now let us move on to the protein classification. The proteins are cellular macromolecules and most abundant, constituting 60% of the dry weight of the cells. They consist of one or more polypeptide chains and has a molecular weight greater than 10,000. They have a unique and diverse structure. The polypeptide chains fold more or less heavily on themselves, inducing a large number of the different three-dimensional structures. It is this that is causing a variety of function, allowing them to be responsible for thousands of reactions in a different cell. Their name, nanomachines, cell is thus justified. For wide variety of cellular responses, we easily imagine that the number of different proteins known to date is very important. It is around 60,000 different proteins are known till today. The classification of proteins, the proteins can be classified based on the structure. Uh, they are classification based on the structure, uh, fibrous protein, globular protein, and intermediate proteins. Classification based on composition, simple proteins and conjugated proteins. And classification based on the function, they can be enzymes, they can be hormones, and they can be transport proteins, they can be toxins, or they can be storage proteins, etc. Right? Okay. So this is a much detailed version of protein classification. So proteins can be classified as simple proteins, uh, conjugated proteins, and derived proteins. So simple proteins can be globular proteins or scleroproteins. So globular proteins are your albumins, globulins, glutealins, prolamines, histones, globins, and protamines. And scleroproteins are, these are non-soluble proteins. So globular proteins are soluble proteins, and these are non-soluble proteins. They can be collagen, they can be elastins, and they can be keratins. So collagen, which is an abundant protein, it is the cementing material. Keratin is present in nails and horns, and elastin is present in the connective tissue. So similarly, albumin is present in the egg albumin. Globulins are also present of the in the hemoglobin. Histones are present in our DNA, which are positively charged. So similarly, conjugated proteins are uh, they contain a non-protein group. They contain a non-protein group along with the protein group. Say example, nucleoprotein, uh, and lipoprotein, phosphoprotein, chromoprotein, metalloproteins. So here we have glycoprotein. So here we have nucleic acid moiety associated with the protein. We may have carbohydrate moiety associated with the protein, lipid associated with the protein, phosphate group associated with the protein. Uh, we have metal group associated with the protein, and all these things they come under the conjugated proteins. And the third one, what we have is a derived protein. So derived are nothing but a denatured or degraded product of simple protein or conjugated protein. So these are nothing but the degraded or denatured. So these are broken proteins, right? When the proteins are broken, we get derived protein either from the simple or the conjugated protein, or in simple term we can call them as first hydrolytic products. So these derived proteins can be primary and they can be secondary. They can be, the first hydrolytic products are called as primary, progressive hydrolytic products are called as secondary. The primary derived proteins are coagulated proteins, proteins, and metaproteins. And the progressive hydrolytic products, the secondary proteins, are the proteoses, peptones, polypeptides, and peptides. So this is the overall summary of the classification of the proteins depending upon the solubility, depending upon the non-protein group, and depending upon the degradation or denaturation of the simple or the conjugated protein. So this is some more information related to the conjugate protein and derived protein. And in conclusion, proteins are high molecular weight biological molecules. Classification of protein is based on the structure, composition, and function and solubility. So again, this is being shown here: simple proteins, conjugated proteins, and derived proteins. So with this, I stop here. Thank you one and all.